Hello students, welcome to Raaonline. Today's chapter is plant growth and development. Plant growth and development under the section of question based discussion of NEET 2020-21 series. And briefing of chapter with the questions is also there. Question number 1 related with the growth. Question number 1 concerned with growth. First one, what is growth? Growth, that is permanent and irreversible increase. Permanent and irreversible increase in size, shape, volume, and dry weight. That is permanent and irreversible increase. Permanent and irreversible increase. That is termed as growth. Growth is non-defining feature. Growth is non-defining feature of living being. That is, if you consult the chapter 1 of 11th class, that is living world, in that case it is given, that is non-defining feature. Why non-defining? Because non-living articles also grow, though their growth is in extrinsic. Growth in non-living being, growth in non-living is extrinsic. They exhibit extrinsic growth, that is only on the outer surface, and that too by external environment. That too is also by external environment. Whereas in growth in living being, that is intrinsic. Growth is performed by every living being. Unicellular organism, they grow by cell division. That is, if number of cell increases, we can say the growth of unicelled organism. So in unicelled organism, growth and reproduction mutually inclusive property. For unicellular organism, growth and reproduction mutually inclusive. For multicelled organism, mutually exclusive. Growth is performed by the plants as well as animal cell. But in animal cell, it is diffused and limited. Growth is shown by animal cell. Growth of animal, that is diffuse and limited. Growth in animal cell, that is diffused and limited. Whereas in plant cell, then this is the question actually. In plants, in plants, the growth is localized and indeterminate. Localized, why it is localized? Actually, in plants, growth is due to meristematic cells. In plants, growth is due to meristematic cell. And meristematic cells are found in growing areas of plants only. That's why localized. Growth in plants is due to meristem. The tissue comprises of living cells. They possess power of division and they are remain in the state of division. This is the property of meristem. And the meristematic cells, they are found in the certain areas of plant, and that area constitute growing region. So that is due to the particular position of meristematic tissue in plant, that's what is localized. Then meristem is present till the end of the life of plant, so that the growth is indeterminate or unlimited in plants. That's why the answer is 4. That's why the answer is 4. Next question, that is second one. The given figure show the development of embryo. You see this is zygote. Zygote divides, two cells are formed. Both these two cells are divide in the state of division. 4 cells. Initially zygote is single celled, then it becomes 2, 2 cells become 4, 4 becomes 8, 8 becomes 16. In initial development of initial development of embryo from zygote, that is geometric growth is seen. In that case, geometric growth is seen. That is part A. Part A, that is every cell are in the state of division. Part A includes those cells they are in the state of division. That is, one cell divides to form 2 cells. Those 2 cells divide to form 4 cells. This pattern of growth is termed as geometric growth. But after that, that is, suppose this is one cell, it divides to form 2 cells. It divides to form 2 cells, out of which one lost the power of division. Then this cell divides to form one more cell. This cell divides to form one more cell, and again a cell loses the form of power of growth. That means there is a constant increase and definite way of a growth, that is, every time one cell divides to form 2 cells, but out of which one cell loses power of division. This pattern of growth is termed as arithmetic growth. Hence, at the time of development of embryo from zygote, at the time of development of embryo from zygote, initially, that is part A, geometric growth is seen, followed by the arithmetic. So the answer is 1. Answer is 1 for that. And come to the next one. Third, this is the graphic representation of arithmetic as well as geometric growth. In arithmetic growth, there is a definite pattern. So the graph is linear. Here, initially the number of cells is 1, then it forms 2 cells, then these 2 cells divide to form 4 cells. That is, gradually number increases and each cell in the state of division. Such pattern of growth is termed as geometric growth. So A is showing when linear curve is there, linear curve is obtained. In that case, the growth is arithmetic, and in this case, the growth is geometric. That is sigmoid curve. Second case shows sigmoid curve. In second case, the sigmoid curve is obtained, and sigmoid curve is seen in geometric growth. That is, initially lag phase, then log phase, then exponential, then growth rate declines. That growth rate declines because of the availability of food, space, and all. That's why the growth phase declines after a certain time, then it becomes stationary. Question number 4. Aquatic plant Ranunculus flabellifer. There are two types of plasticity, that is adaptations. That is, it is with regards to the development and with regards to the environment. And Ranunculus flabellifer, that is commonly known as buttercup, its submerged leaves are highly dissected, whereas emerged leaves are broad and lobed. That is, this is because of the habitat. This is because of the habitat or environment in which it lives. Habitat or environment it is in live. So this is different types of presence of different types of leaves, that is heterophily, and it is with regards to the environment both. So here this answer is that is, both are correct, heterophily and environmental plasticity. So my answer is D. That is, In aquatic environment, leaves are dissected, whereas in normal environment, immersed leaves are broad and lobed leaves. This is good example of developmental plasticity. Formation of interfascicular cambium and cork cambium in dicot stem. These are the three terms related with the division. Suppose this is a cell. This is a cell, and this cell divides and two cells are formed, and out of which one cell loses the power of division. That is undergoes differentiation. This is undergoes differentiation. The second one, dedifferentiation, that is this cell. If this cell regain the power of division, if this cell regain the power of division, it is termed as dedifferentiation. And out of these two cells, suppose this cell loses the power of division again, that is redifferentiation. That is redifferentiation. If a cell divides and produce a cell, and that cell will lose the power of division, it get differentiated. If it regains the power of division, get dedifferentiated. Then after dedifferentiation, definitely it will divide. And obtained cells, if they lose their power of division, if they stop their division, then become redifferentiated. That is redifferentiation. So interfascicular cambium and cork cambium. Interfascicular cambium, see the sectional diagram of dicot stem. In dicot stem, vascular bundles are arranged in a ring. Vascular bundles are arranged in a ring. And these are the two vascular bundles. These are the two vascular bundles. Vascular bundles are arranged in a ring, and between the primary xylem and primary phloem cambium is present. But this is fascicular or vascular cambium. The cambium present between primary xylem and primary phloem. Cambium present in between primary xylem and primary phloem is, that is fascicular cambium or vascular cambium. Fascicular or vascular cambium. In between them, here medullary rays are present. Here medullary rays are present. In between them, medullary rays are present. These are parenchymatous. These are parenchymatous medullary rays, and power of division is lost in them. Power of division is lost in them. These are medullary rays. Cells of medullary rays. At the time of secondary growth, the cells of medullary rays, these cells regain the power of division. That cells regain the power of division. The power of division is already lost in them. That is, they are differentiated. But at the time of secondary growth, they regain the power of division and they start their division. Hence, my answer is good example of dedifferentiation. So let's make the another diagram in this support. That is vascular bundle. This is also vascular bundle. In between two vascular bundle, in a vascular bundle, fascicular cambium is present. This is fascicular cambium or vascular cambium between primary xylem and primary phloem. And you see, these cells regain the power of division. These cells regain the power of division. And they become meristematic. So, this cambium which is present, this is actually fascicular or vascular cambium. This is fascicular cambium or vascular cambium present between primary xylem and primary phloem, and this is interfascicular cambium. This is interfascicular cambium, and it is formed at the time of secondary growth only, and it develops from the cells of medullary rays which are parenchymatous. They already lost their power of division, but at the time of secondary growth they regain the power of division and become meristematic. So power of division is regained, hence this is dedifferentiation. And similarly cork cambium. Cork cambium is present. Cork cambium is not there initially in primary structure. This is the epidermal region of dicot stem. This is epidermal region of dicot stem. And this is epiderm. And just below the epiderm cortex is present. This is cuticle. This is cuticle. And just below the epiderm cortex is present and made up of parenchyma. This cortex is present, made up of, upper few layers collenchymatous followed by parenchymatous. Upper few layers collenchymatous, then other layers are of collenchymatous in the cortical region, and both are non-dividing cells. Both are non-dividing cell but living. At the time of secondary growth, in cortical region, few cells regain the power of division. Few cells of cortex regain the power of division. Few cells of cortex regain the power of division and they constitute cork cambium. So cork cambium is formed due to the division in permanent living cell. Due to the division in permanent living cell. This is cork cambium. Or phallogen. And phallogen is formed from the differentiated cell. That is, differentiated cell regains the power of division, hence its origin is also good example of dedifferentiation. Cork cambium, it appears in which region of dicot stem? That is in cortical region. It is from the cortical cells which are living and permanent and they are non-dividing. They regain the power of division. That's why they are the example of dedifferentiation. In root, if it is in root, if we say about the root, dicot root, that is cambium as well as entire cambium as well as cork cambium, both are secondary in origin. So this is the answer of question number 5. Now heterophyly found in all plants given below except cotton, coriander, larkspur. They exhibit developmental plasticity. That is, during juvenile stage the shape of leaf is something different, and in adult phase the leaf of the plant is different. So at the time of development, two different types of leaves are seen in juvenile stage as well as in adult phase. So these three are the example of developmental plasticity. These three are the example of developmental plasticity. Hence answer 4th is mismatch plasticity. Developmental plasticity. That's why the answer is 4th one. Here first three are alike, but the last one is different. So this is the correct answer here. Exponential phase, that is maximum growth. Exponential, that is reach to the maxima. Growth slow down and accumulates toxin. No. growth progresses rapidly, reach its maxima. So this is the exponential phase. This is the exponential phase. After that, the growth will slow down. After that, the growth will slow down. Terpenes, actually every hormone of plant definitely having some chemical nature. For example, ethylene is C2H4, it's a gaseous hormone. Ethylene is gaseous hormone and it is formed from the methionine amino acid. Uh, this is adenine derivative. This is adenine derivative. Purine base adenine derivative. Purine base adenine derivative. Gibberellins are terpenes. Gibberellins are terpenes. Gibberellins are terpenes. Whereas IAA, indole 3 acetic acid, these are all indole compound. Oxins are indole compound. So that's why the answer is gibberellin. Gibberellins are terpenes. Now come to the ninth question. The pair that consists of plant growth promoters only. Actually, plant growth regulators are kept under two categories. One promoter, two inhibitor. In this series, let's know auxin is promoter, cytokinin is promoter. This is promoter, this is also promoter. Gibberellin is promoter, but this is natural growth inhibitor. ABA natural growth inhibitor. Ethylene is in both categories, that is promoter as well as inhibitor both, and this is inhibitor only. So my answer for this, that is which pair that consists of growth promoters only. Correct answer is A. Correct answer is A. This is natural growth inhibitor abscisic acid, and this is kept under both the category, that is some functions are promotional and others are inhibitional. Match the column. Natural auxin is indole 3 acetic acid, that is for A with 3. A with 3. Natural auxin indole 3 acetic acid. Then come to the second one, synthetic auxin. Synthetic auxin is the first one, naphthalene acetic acid, alpha naphthalene acetic acid, that is B with 1, B with 1. And other natural auxins, other auxins which are synthetic, that is 2,4-D also. One more example you can add 2,4-D. It is also synthetic auxin, that is 2,4-dichlorophenolic acid, that is selective VD side. This is selective VD side. Selective VD side. Then Bakani disease is concerned with the rise, and it is in Japan, and it is with the discovery of gibberellin. Actually, Japanese farmers while working in paddy field, they observed abnormal tallness in paddy seedling. They called them Bakani. Bakani is a abusive language in Japan, and it is foolish seedling disease. That is foolish seedling disease which leads to the discovery of gibberellin hormone. Finally, this Bakani disease of rice leads to the discovery of gibberellin hormones. Some scientists observed a fungus in the neighborhood of those abnormally tall paddy seedlings, and that fungus was Gibberella fusicori. Gibberella fusicori. And the secretions of that fungus were responsible for that abnormal tallness. That hormone is later recognized as gibberellin. Gibberellins are gibberellic acid. Gibberellins are gibberellic acid. They are acidic in nature in aqueous solution. Then come to the last one, natural cytokinin, that is GATN. Natural cytokinin is GATN, that is with D option we can give the second one. So choose with this respect. 314, 314. This is 3, this is 3, so this is automatic rejection. These two are not possible with rejection method. Then come to these one, that is 3142. 3142. The correct option is A. Correct option is A. Cytokinin, natural cytokinins, they can also be seen in, that is immature, extract of immature maize grain, extract of immature maize grain, immature maize grain is an example of cytokinin. And coconut milk. Coconut milk also possesses this particular property. Coconut milk or coconut water. These two are zeatin category, that is naturally occurring cytokinins. Now come to the next question, that is 11th question. Gibberellic acid increase yield of sugarcane. It is right match because gibberellin is known for internode elongation. This is rightly matched. Gibberellin, if we apply gibberellin even on genetically dwarf plant, they become plants of normal height. They become plants of normal height. If we add gibberellins, over the genetically dwarf plant, even they become plants of normal height. But that for that particular generation only, because gibberellin induces the internode elongation. Gibberellin, suppose this is a plant and these are the node, and in between two nodes this is internode. If we apply gibberellin, you see, that is, this is the striking effect of gibberellin, that is elongation of internode. Cytokinin promotes apical dominance. This is wrongly matched because cytokinin actually counteracts. Apical dominance, and apical dominance means there are two types of buds in plant. This is apical bud found in the apical region, and this is lateral bud in the axis of leaf. Generally, if apical bud is there, the growth of lateral bud suppresses or inhibited. This is called apical dominance, and apical dominance is due to auxin. Apical dominance is due to auxin. Apical dominance is due to auxin, and counteraction of apical dominant. Counteraction of apical dominance, AD. Apical dominance. This is the role of cytokinin. That is anti-auxin like activity. So definitely the answer 2 is wrong. Mismatched. Ethylene responsible for sprouting of potato. That is potato sprouting, that is on potato. This is potato tuber, and these are eyes over potato. And in the eyes, lateral buds are present or axillary buds are present, and ethylene induces its sprouting. That Then last one, ABA, natural growth inhibitor. This is natural growth inhibitor. Natural growth inhibitor, ABA. It is also called dormin because it promotes the dormancy of buds and seed. So, but natural inhibits seed germination. But natural inhibits seed germination. Question number 12.